Today I'd like to begin a discussion of systematics as it relates to the greater Yellowstone ecosystem. And so systematics is the study of the diversity of life, both past and present, and the relationship of organisms through time. And as part of that, taxonomy is a part of that. Taxonomy is the science of classification. And as we consider classification, what we're really going to be interested in is what's known as phylogenetics. And in phylogenetics, it's all about establishing the evolutionary relationships of organisms through an approach known as cladistics, which I'll be discussing later. So when we're classifying, I want to go through kind of a quick overview of some of the kinds of characteristics that we look at in trying to establish the evolutionary relationships in organisms. And certainly this begins with genetic analysis, because as we've established, if organisms have a common ancestry, they share genes. And so we look at DNA and RNA of organisms and try to get a sense of the degree of relatedness between them by looking at that genetic material. And as an indirect measure of looking at genetic traits, we will look at other traits, and those include morphology. Morphology is the physical or a study of the physical structure of organisms. And when we consider morphology, we'll look at what we call fine anatomy or fine morphology, and that would be the microscopic anatomy of organisms. So we'd be interested in the structure and types of cells of which they're composed, tissues of which they're composed, that kind of thing. We will also consider the gross anatomy of organisms. So this would be the macroscopic features of organisms. So we might look at invertebrates, we might look at the skull anatomy and skeletal anatomy. We would also look at the organs that they possess and the nature and types of those organs within the organism. We would also look at the biochemistry of organisms. This is especially true when we're looking at small organisms, microorganisms. But we certainly look at the physical composition, biochemical composition of organisms. We're also interested a lot of times in the biochemical pathways exhibited by organisms. What kind of biochemistry do they exhibit in terms of pathways? And we've talked about heterotrophs, for example, before. Heterotrophs would be organisms like you and I that must ingest organic molecules because we cannot make organic molecules from inorganic sources. And then another example of this would be autotrophs. Autotrophs can make organic molecules from inorganic sources, and we've talked about this before. So they can take inorganic molecules, they'll use some source of energy and put those together to form organic molecules. And we talked about this in photosynthesis. Carbon dioxide and water combine to form glucose, sugar. So what I don't think we talked about, though, are the types of autotrophs that we can have in nature. Some autotrophs, like those that practice photosynthesis, are photoautotrophs. So they'll be using light energy to put those inorganic molecules together. And then there are some organisms that we refer to as chemoautotrophs, and the chemoautotrophs will carry out inorganic chemical reactions, utilize that energy to construct organic molecules. And so this is typical of some types of bacteria and archaea that we'll be talking about. Now, continuing this idea of biochemistry, other characteristics that we would look at, biochemical pathways that can be important. One is oxygen toxicity or oxygen tolerance, various ways to describe it. Some organisms, like you and I, are described as obligate aerobes. And so the obligate part of this, if you're obliged to do something, it means you have to do it. And in this case, aerobes is referring to air, but specifically oxygen. So if you are an obligate aerobe, that means you have to have oxygen. So certainly animals would fall into that category of being obligate aerobes. You might be surprised to know that there are some organisms actually that are obligate anaerobes. And so the prefix an means without. So there are some organisms that really cannot tolerate oxygen. They must be in environments without oxygen. And living in our gut, your gut and my gut, as well as in the gut of a lot of organisms in the Yellowstone ecosystem, there are many bacteria and archaeans that are obligate anaerobes. They actually cannot tolerate oxygen. And then there's still other organisms that are facultative anaerobes. They can live in environments without oxygen. They can also live in environments with oxygen. There are many fungi that fall into this category, and an example would be yeasts. Yeasts in an anaerobic condition will form alcohol, ethyl alcohol, but they can also live in aerobic conditions. Other characteristics we'll look at is embryonic development. A lot of times by looking at the adult organism, they may not look very much alike. You may not think they're closely related to one another, yet when you look at their embryology, you see that there are a lot of similarities there. And so this will be something that we consider down the line, and we'll talk about specific examples, but it's certainly something that helps you establish evolutionary relationships. Also looking at the life cycles of organisms, and this can include embryonic development, but here we're looking at modes of reproduction. How do they reproduce? We're looking at metamorphoses. In other words, do they go through a gross physical change? And if they do, what type of physical changes do they go through? We're kind of used to our own development, our own life cycles, where we're born as little humans and we develop into big humans. But that's not always the case. In fact, that's really not typical out there sometimes in nature. And so we'll look at situations where we see gross changes, physical changes, as organisms go through their development. Also, we may look at, if it's a parasitic organism, what hosts does it use? That might be something that we look at, among other kinds of factors in life cycles. And then we may also consider the ecological role of organisms in giving us clues, trying to ascertain clues in relatedness. Is the animal a carnivore? Is it an herbivore? Is it an omnivore? That also might help us determine relationships. But ultimately in classification, it's all about establishing evolutionary relationships, and it's not always who looks alike that tells you that, believe it or not. So especially when we look at adult organisms. Now, in considering classification, we're going to be using what's known as the Linnaean system of classification, and the father of the Linnaean system was a man named Carl von Linné. And he's the person who's responsible for Latinizing the sciences. He wanted a common language so that the European scientists could communicate with one another. And he was such a fanatic about it that he actually Latinized his own name to Carolus Linnaeus. So we consider Linnaeus then to be the father of classification. Now, in the Linnaean scheme, he had kind of a nested box kind of approach. This was purely an organizational approach rather than an evolutionary approach to classification. So the Linnaean approach, Linnaeus was not an evolutionist. He didn't know much about evolution. This was really an organizational tool. And in his approach then, like I said, it's kind of a nested box kind of approach, and it started with a category known as the kingdom, and the kingdom would be the broadest, most heterogeneous, most mixed group of organisms. And so within a kingdom then you could divide that into organisms that were more alike physically, and he was looking mostly at physical characteristics and primarily plants. And so a kingdom can then be divided up into subcategories or subgroups called phyla or divisions, and those terms are synonymous, so we can use those interchangeably. And then phyla or divisions can be broken up into groups that are more alike, which would be classes. Within the class, you could have groups that are still more similar to one another, which would be orders. Orders could be broken up into families. And then families can be broken up into genera. Genera is the plural of genus. And then a genus can be broken up into species. And so species then would be the most homogeneous group, like organisms, and we've talked about that before. Generally, we define that as being organisms that will successfully mate with one another. Now, if we apply that to an example from the Yellowstone ecosystem, if we look at the wolf then would be in the kingdom Metazoa. That would be the animal kingdom. They belong to the phylum Chordata, organisms that have a dorsal hollow nerve cord and some other characteristics. Mammals, so they have mammary glands, fur, hair, warm-blooded, among other characteristics. The carnivora, so special types of teeth related to feeding on meat and other skull and skeletal characteristics. The canids would be the dogs. The genus also is a group within the dogs, very closely related to our domestic dog. And then the species then would be Canis lupus. So this is also an invention of Linnaeus, is generating this Latin scientific name for organisms. And so to generate a scientific name, you take the genus name and the species name and put those together. So in the case of the wolf then, the genus is Canis, species is lupus, so scientific name Canis lupus. Now, as we've evolved in a sense in classification, the Linnaean system creates some problems. Again, there's only the 6 or 7 categories we had there, and evolution does not always proceed in a precise number of steps. And so we've had to add categories over time. And just to give you an example of the kinds of additions we've made, for example, now there's a much larger grouping than kingdom known as the domain, and we can divide domains into subkingdoms, or excuse me, subdomains, and subdomains into superkingdoms, and superkingdoms into kingdoms, and kingdoms into subkingdoms, and subkingdoms into branch into infrakingdoms, and so on. So we've invented a lot of new categories to try to accurately reflect the many steps that may occur in an evolutionary process. So I'll try to stay away from a lot of these terms, but we are going to have to worry about some of these taxonomic groupings, but we'll get into that in more detail later. Now, in a phylogenetic approach to classification, our goal is to establish what we call monophyletic groups. And so a monophyletic group would be a taxonomic grouping, also called a clade, where the members trace back to the same common ancestor. So again, a monophyletic group would be composed of individuals that trace back to the same common ancestor. And we refer to a monophyletic group as a clade, C-L-A-D-E. So let's look at an example. What do I mean by that? Well, let's say we have a number of species here, A, B, C, D, E, F, G, and H. And let's say that A and B trace back to a common ancestor I, C and D trace back to a common ancestor J, E and F trace back to a common ancestor K, G and H common ancestor L, I and J trace back to a common ancestor M, K and L common ancestor N, and then M and N both trace back to a common ancestor O. So at some point they're all going to trace back to the same common ancestor, but again, we'll see how we would apply this to monophily. So again, these would constitute our different species. So let's say then that we want to establish some groupings here. We want to do some classification here. So in other words, if we took A and B both trace back to common ancestor I, so that if these are species, then we could have a genus I that includes A and B, species A and B. We could have a genus J that includes species C and D. We could have a genus K that includes E and F, and a genus L that includes species G and H. So again, each of these would be monophyletic groupings because the members trace back to the same common ancestor. Furthermore, we could look at what individuals trace back to M, and we could generate another clade there. So this would be another monophyletic clade. Again, these four species all trace back to the same common ancestor. So that would then be a different group. So we'd go from genus to family here. So it would be a family M. And we could create a family N composed of these individuals that trace back. And so on. So as long as all members in a group trace back to the same common ancestor, then that's a monophyletic group. Now, there are two conditions that we want to try to avoid. One of these is what's known as polyphily. So in polyphily, you're including individuals that really should not belong in the group, individuals that do not trace back to the same common ancestor. And in paraphily, you're excluding individuals that should be included in your group. So again, taking an example here, again, let's say that we have basically the same relationships. If we looked at a genus here, if we created a genus where we had species E, F, and G included, that would be a polyphyletic group. In other words, G is not tracing back to K. So G does not have the same common ancestor as E and F. So this is an inappropriate grouping. This is a polyphyletic group. And then conversely, if we had a genus L that contained only species H, that's a paraphyletic group. We've excluded G, which should trace back to the same common ancestor, and so we've excluded a member, so that would be a paraphyletic group. So both of these are mistakes that we want to try to avoid in our classification. We don't always avoid them, but we make every attempt to avoid that. And again, this would be another example if we went for a larger grouping here. If we looked at a family M, we're including individuals that do not trace back to the same common ancestor, so that would constitute a polyphyletic group. And this would constitute a paraphyletic group because we've omitted individuals that should be included in that taxonomic grouping. So again, one of the dilemmas then is establishing the relationships. And so historically, we relied on appearance. But appearance is not always the best way to establish evolutionary relationships. And just to give you an example from my family, these are actually my daughters. This is Valerie and Melanie. And they may look somewhat alike, but they have quite a few differences, actually. Valerie's much lighter skinned, blue eyes. Melanie has a darker complexion and brown eyes. And so they kind of look like sisters, but they don't look a lot like sisters. And again, if we kind of expanded this out, if we looked at Melanie and a cousin that they have named Christopher and Valerie, if you're just basing this on appearances, Melanie and Christopher look almost exactly alike. They have, again, the same coloration, facial shape, and so on. I mean, they really look like they're closely related. So if you were building a family tree based on appearances, you'd say, gosh, Christopher and Melanie look like their brother and sister, and Valerie must be a more distant relative. Maybe she's a cousin or something like that. But of course, that would not accurately reflect the family relationship. There you're grouping according to appearance, but appearance is not necessarily telling you the relationships. Because the actual relationship, the familial relationship, the genetic relationship, would be that Melanie and Valerie should be grouped together. So this is one of the problems that we have in classification, is that if we rely too heavily on just appearances, kind of general characteristics, we may not actually be reflecting the evolutionary relationship, which is similar to the family relationship that we just went through. So kind of this historical approach based primarily on physical characteristics is the traditional approach. And so the traditional approach, you will just lump according to similarities, you'll separate out according to differences. But there's no particular system to this approach. And so let's take an example. Let's take a couple of 3 different animals that are really not that closely related, but we'll just kind of use this as an example. So let's say we have a trout, a lungfish, and a cow. And let's say that we were trying to establish relationships here. In the traditional approach, you would just be lumping according to similarities, separating according to differences. And again, this would seem pretty obvious. Okay, fish are fish. We'll lump the fish together. The cow is nothing like a fish, and so that would be in the out group. And so if you were establishing a family tree here, or what we call a cladogram, you'd more than likely set it up this way. I'm going to lump my fish together, and the cow is the out group. Cow doesn't belong with fish, so this would constitute a group. The cow would constitute some other group, not as closely related. Now, we could take another approach, though. And this is known as the cladistic approach. And this is the modern approach to classification. In the cladistic approach, we try to establish what are known as ancestral characteristics. We try to look at characteristics and decide whether they recently evolved or more distantly evolved. And the ancestral characteristics would be the ancient characteristics, those that evolved a long time ago. And we also try to look at characteristics that have more recently evolved. We call these the derived characteristics. Individuals that share recently evolved characteristics then would have to have a recent common ancestry. If they share ancient characteristics, that would be an ancient relationship. Recently evolved characteristics would show us a recent evolutionary relationship. So again, we can look at the fossil record. There's some other mechanisms to do this, but we try to establish which are ancestral and which are derived characteristics. And so in this kind of approach, again, it's not just a matter of lumping and separating. We'd be interested in ancestral and derived characteristics. So in this case, certainly evolution of the lung would be a derived characteristic compared to the gill. And what distinguishes the lungfish from other fish is that they do produce a lung-like structure off of their esophagus, and they gulp air. They actually have to augment what they get from the water with air, oxygen from air. And let's take this a step further. Let's say that you have found the developmental gene for lungs in cows, and when you look at the lungfish genome, you find that same gene in the lungfish. So they actually share a gene that leads to development of this rudimentary lung and eventually a more refined lung in cows. They share that gene, but that gene is missing in the trout. So in other words, you've identified a very specific derived gene. So in the cladistic approach then, because these two share a more recently evolved gene and share a more recently evolved physical characteristic, these have a more recent common ancestor. So in the cladistic approach then, we would form a cladogram that looks like this. And so this is very counterintuitive. This doesn't feel good. You mean to tell me you're telling me a lungfish is more closely related to a cow than it is to a trout? And the answer is yeah. They have a more recent common ancestor than either does to the trout. And so this would be the appropriate diagram to reflect an evolutionary relationship as opposed to just an organizational relationship. So the cladistic approach, again, can be a little counterintuitive, but we find that this is the best approach. Another example, looking at our own history, is that if we looked at apes like the gorilla, and also looked at an ancient organism known as the Australopithecus and then compared that to modern humans, Homo. When you look at Australopithecus, again, Australopithecus shares a number of derived characteristics with modern humans. These are related to upright walking. Gorillas are knuckle walkers. Upright walking came much later. And so when you look at the skeletal anatomy of Australopithecus, as you look at the foot structure, lower leg structure, structure of the upper leg, the hips, these are animals that were adapted for upright walking. And so again, when we build our cladogram, then we would link the Australopithecus to humans rather than to the gorilla. We share a more recent common ancestor with them than they do with gorillas or chimpanzees or any other animal. So again, this accurately reflects evolutionary relationships. Now, as we look then at nature and the groups of organisms we find in nature, we look at 3 domains of life. And this was proposed back in the 1970s by a researcher named Carl Woese. And so he proposed 3 domains of life: the eubacteria, the archaea, and the eukarya. And the eubacteria and the archaea, what they have in common is that they are prokaryotic organisms. And you may recall the prokaryotic cell is a simple cell, small, lacks a true nucleus, and so on. The eukarya are composed of all of the eukaryotic organisms. And so he, again, saw nature as being composed of these 3 distinct groups. And so plants and animals would be an example of eukaryotic cells. And you may recall that eukaryotic cells, have a membrane-bound nucleus. They have a number of additional organelles. So these are large, complex cells. So we're going to be examining these 3 domains in some detail, but just at first looking at some of the general characteristics of the 3 domains and then establishing which domain came first, because Woese's position was that the archaea were the ancient group, because many of the members of the archaea are found in extreme environments. We refer to them as extremophiles. So anyway, as we began to learn more about these different types of organisms, we wanted to come back and look at that supposition that the archaeans were the most ancient group and see if that held up over time. So the bacteria, or eubacteria then, the domain bacteria or eubacteria. What makes bacteria unique? And there are a number of characteristics. This is kind of a complex chart. We're not going to worry too much about the details here. I'm just going to summarize some of the important characteristics. But when we look at the eubacteria and what makes a eubacterian, we're going to focus on the cell wall. The bacteria have distinctive cell walls, and in particular, they have a chemical known as peptidoglycans in their cell walls. So that is a distinctive characteristic for the bacteria. Now, when we look at the archaea, a distinctive characteristic for the archaea is that they have very unique plasma membranes, unlike other organisms. And so what is unique and interesting about their plasma membranes, you may recall that plasma membranes are composed of a biphospholipid layer, I should say, and the phospholipids have a water-soluble head and water-insoluble tails composed of fatty acids. And so in the archaeans, their fatty acids are kind of unusual because most fatty acids are just single chains, or what we call straight chains of carbons, and they have branching chains of carbons in their phospholipids. And they also have what we call transmembrane phospholipids. Some of their phospholipids go all the way across the membrane instead of being in 2 layers. So anyway, that's very unusual, and the archaeans exhibit those physical characteristics. And then the eukarya, what makes the eukarya unique? Well, what makes the eukarya unique is that they are eukaryotes. So they have membrane-bound nucleus, membrane-bound organelles, complex cells. So then we begin to look at, well, what are shared characteristics? And so when you look at the bacteria and the archaea, they do share some characteristics. And some of the characteristics that they share is that they lack a nuclear envelope of any kind. They lack membrane-bound organelles. They're both very small. And they also have a circular chromosome. They also will have smaller loops of DNA called plasmids. So these are all prokaryotic characteristics. They both have prokaryotic ribosomes, for example, as well. So they're basically prokaryotic cells. So that's what they have in common. And prokaryotic characteristics are ancestral characteristics. The first organisms to evolve were prokaryotes around 3.5 billion years ago. Now, as we look at shared characteristics in between archaea and eukarya, we do have some there as well. And when we look at the archaea and the eukarya, Some of the characteristics that they share is that they both lack peptidoglycans in the cell wall. They have similar types of RNA polymerase. They always use the first amino acid, which is methionine, and when they're building proteins. They have histones associated with their DNA. They are antibiotic resistant, and these are considered derived characteristics. We think these are more recently evolved characteristics because we associate them with eukaryotes, yet archaeans show some of these characteristics as well. So if we were to take the traditional approach, as Woese did, then you would look at these three groups, the eubacteria, the archaea, and the eukarya, and you'd say, well, the prokaryotes are basically more alike, so they must be more closely related. You'd probably lump them together, and the eukarya would be kind of the outgroup. But in the cladistic approach, we're going to come to a different conclusion. One is that the eubacteria, archaea, and eukarya then, if we look at this relationship, is that we would end up putting the archaea and the eukarya together. And again, it's because they share derived characteristics. They share more recently evolved characteristics, so we think that this is the accurate evolutionary relationship. So we think that Woese was wrong. Even though archaeans tend to like these extreme environments that Woese thought was similar to the early Earth, we really do think that the eubacteria came first, then the archaea, and eventually the eukarya, and we think that this is the relationship here. Now, when we're looking at these very, very ancient relationships, again, which trace back billions of years, 3.5 billion years, 2.5 billion years if we're talking about the eukaryotes, it's complex, and we're going to talk more about this in a future module, because we not only have the archaeans and eubacteria that can transfer genes from generation to generation, but they also exchange genes with one another. And so the evolutionary relationships get a little muddled here. And so as we look at this, what we actually think is going on is that there's evidence that archaeans and eukarya have assimilated to actually form eukaryotes. Because certainly eukaryotes have assimilated some eubacterians. There's evidence that they came from the archaea. So we're still looking at what happened in this relationship. There's evidence that the eukarya are kind of an assemblage of both archaeans and eubacteria. So this might be a better way to describe the relationship between the three groups. So as we move forward then, we're going to begin to look at these prokaryotic domains, the eubacteria and the archaea, and we'll want to look at examples specific to the Yellowstone ecosystem, and from there we'll begin to move into the eukaryotic organisms, again looking at organisms specific to the Yellowstone ecosystem. So that's it for today. I'll see you next time.